Okay, so this is going to be a mixed media presentation with all the confusion that it's going to cause. But part of the issue is I was planning to just do a Blackboard presentation, but not finding a Blackboard and seeing a whiteboard instead, I got a little worried and migrated some of the stuff onto the slides. And so I'm going to start with the slides and then going to switch to the Blackboard, which is the whiteboard. And if you can't see it, then please come forward. And if not, never mind, just ask questions and we'll handle it. So, oops, I just made a mistake. Sorry, just a second. So far, so good. So I'm going to talk about statistical genetics, which at this stage shouldn't be very clear exactly what it is. But I hope in the next hour, it will come into focus. And if it doesn't, ask me. And in any case, just keep interrupting and keep asking. This is the only way I know that I'm in touch with you. So, but statistical genetics is all an attempt to understand the dynamics of evolution. And very often when we, before I do this, never mind the outline. So it's very natural, of course, for everybody, and certainly for the physicists, to think about evolution because, like Mukund today was talking about, evolution, trying to understand how the cell has come to be, how eukaryotic cell has come to be what it is, and it's really only possible to understand this in the light of evolution. And then, of course, when we think about evolution, we often think, just like Mukund was doing in his lecture, in the context of sort of the grand scheme of things on the scale of billions of years, and thinking about the origins, thinking about the tree of life. And the thing with the tree of life is that we can only observe its leaves. And again, Mukund explained it very well. Among these leaves, we're located actually very close to amphibians, and in fact very close to yeast. And if we want to find the common ancestor, we have to go into a geological record and dig for fossils. So we will be concerned here with the dynamics of evolution. And dynamics is a continuous process. It's happening all the time. It's happening now. And to understand what this dynamics, we actually have to zoom in here and take this gigantic magnifying glass and look at the variability, both phenotypic and genetic variability, in the single population, be it humans or be it the frogs. And of course, there is also competition between different species, right, in the ecological context. But to the extent that the natural selection is acting on genetic variation, it is acting on little variants which do not show up in this grand scheme of things. So what we will want to do is zoom in and talk about what's happening in the populations. And zooming in was rather more difficult to do 150 years ago when scientists were sort of studying the diversity of life, they had sort of morphological features to go by, right? So that's what the original classification was based on. Now, of course, we have the ability to reconstruct these genealogical trees from DNA sequences themselves. And in fact, that allows us to actually observe evolution in action. So of course, in order to observe evolution in action, you have to look at some critters that are evolving fast enough. And nobody evolves faster than the viruses. And so in particular, these days, we have data on HIV evolution. And it is obtained by isolating viruses from a blood sample. And AIDS is a chronic disease. And at least some of the patients receive decent medical care and are followed through evolution, well, through the unfolding of the disease. So in this case, blood samples were taken, I think, every 6 months. And then the virus was sequenced. And what you see here is an evolutionary tree, or if you like, a genealogical tree of the virus in these samples. And initially, one saw very little variation. So this isn't actually very honest, because you see this -2. So this tree is actually extrapolated backwards in time. So these samples weren't really taken. But nevertheless, early on, there was a fairly little genetic variation among the viruses in the sample. And here, the tree is basically constructed on the basis of pairwise distances. So there is just, well, in fact, there are many canned algorithms that will construct trees if you give them sequences. But the interesting thing here is that what we see are not just the leaves. We see the full dynamics of evolution. We actually see the evolutionary history. And one observes that there was a great deal of diversification early on, and then some of the strains survived, and many died off, and so on and so forth. And very sadly, in the data of this sort, towards the end of the data, one actually sees sort of a collapse of diversity into one narrow clade. And of course, basically what's happening here is the virus is mutating and evolving to beat the immune system, which is also evolving to adapt, or rather to destroy the virus. And the virus is adapting to avoid it. So in the course of 10 years, one sees something like 10% of divergence, so nucleotide by nucleotide. So 10% of the genome got substituted for something else, right? So to see that much divergence in an animal, in Drosophila, would basically take many millions of years. And so here, evolution is really just the course of the disease itself. Now, I already said that this rapid evolution of a virus is driven by selective pressure on the virus to avoid the immune system. So I don't know if you noticed the date on my previous slide. So that data was 15 years ago. So that patient actually was not treated. There was no sort of universal drug treatment at the time. So these days, patients who at least go to the clinic get treatment. Yes, please.
So the last slide, Boris, the mutating lines were the ones that are not in the sample or the ones that are not registered?
Yeah, so those are the ones that were in all samples, but are not most plausible ancestor of the later sample. Yes.
So in this slide, the divergence was mainly in those area of the codes which were resistant to drugs, or the phenotype of the disease itself? What areas were difference in anything or anywhere? Or?
Right. So some areas are much more conserved than other areas. And then there are some genes which basically appear, basically epitopes for the immune response mutate much more rapidly. Yes?
So that earlier in the earlier slide, that's the phylogenetic tree of the genome for the virus. Right? Now, during calculation of the pairwise distance, as you're saying, so it's not only mutation. Sometimes what happens, some shuffling of genes, combinations takes place. So how is that?
Okay. So we can, of course, go into a much greater depth here. So I lied to you. It's not the whole genome. It's actually one gene. And here you're only looking at substitutions. Okay. So now that there is drug treatment, and now, of course, the problem is that HIV very rapidly evolves the drug resistance. And here, just for laughs, I printed out the actually amino acid sequence of the protease gene, which is the target of one of the drugs. And this is the sequence of the same gene from a drug-resistant strain. And in red, if you can see, I marked the amino acid substitutions. And what we know is that there's a whole bunch of substitutions in this resistant strain. And what we know is not all of these substitutions are necessary to evolve drug resistance. But we also know that it takes more than one. It takes multiple substitutions. And this is actually very important because HIV virus is, well, it's an RNA virus, so it has a very high mutation rate. So basically 3 times 10 to the 5 per base pair per generation. The length of HIV genome, I think is 4 times 10 to the 4, right? 40,000, 40 kb. So that basically works out to so few tenths of mutation per genome generation. But the viral population is gigantic. This is the number of viruses in a single patient. So that basically means that in one viral turnover, every single base pair is mutated. Every single pair of base pairs is mutated, right? And the bottom line is, if it only takes a single amino acid substitution to acquire drug resistance, that's all over in 2 days, right? And the difference between a good drug and a bad drug is that a good drug will require multiple mutations, multiple substitution, just to get the task more difficult. And that's the logic behind the drug cocktail. It's not enough to use even one good drug. It's better to use several, right? So that in order to evolve the resistance, now this virus actually has to acquire multiple mutations and traverse some non-trivial fitness landscape. And okay, so we have high mutation rate, very large population, and there is another twist to the evolutionary dynamics of HIV. HIV, it's having its own sex in a very peculiar way. So it's an RNA virus. It has actually two copies of this RNA molecule. And in order to do anything, this virus has to translate RNA into DNA. So it has to reverse transcribe. And it has an enzyme called reverse transcriptase, which reads an RNA copy and makes a DNA of it. So now that enzyme then starts making a DNA copy, but it's a bit sloppy, and it loses its template while holding on to the nascent DNA, right? And then it reloads whatever other template comes by and continues from where it stopped. So now, if there is only one virus present in the cell and both copies are identical, it doesn't matter. It reloads and just continues. However, if the cell is multiply infected by genetically divergent strains of the virus, so now it's going to proceed, so with some relatively significant probability, it's going to reload, so an RNA template of a different variety, and will make a mosaic offspring, right? So there is recombination mediated by a rather unusual mechanism, but with the same effect. So I gave you that very much abused quote of Dobzhansky, that nothing makes sense except in the light of evolution. But Michael Lynch, who is a population geneticist, transformed it into a statement that in order to understand the dynamics of evolution, we actually have to understand population genetics. We have to understand what's happening, right? We have to take this zoom objective and look at what's happening within populations. And the kind of questions we want an answer to, which are very obvious in the context of my HIV example, is, well, how fast is the virus going to acquire drug resistance, right? How does it depend on how many mutations are required? How does it depend on the population size? How does it depend on the viral load, right? What is the role of this viral recombination? What is the role of that process in the rate of evolution, right? In the rate of acquisition of resistance. So we expect that it's going to play possibly a big role in the dependence of that rate on the population size, right? So of course, in a small population, it is unlikely that one cell will be doubly, so multiply infected. But when the population size gets larger, right, then this recombination process will possibly turn on. So we want to understand this. And ultimately, and I guess I was sort of hoping I would also talk about this, not today, but tomorrow. Now I'm not so sure, given the rate at which I'm going. You know, there's sort of an ultimate question. If we think that we understand evolutionary dynamics, can we predict the evolutionary future? And you may say, well, and he's a raving lunatic. How can we don't even predict the weather terribly well. So how are we going to predict that? And I would argue that, you know what? Actually, we can predict weather. It's just that we can only predict it in the short term, right? And the way we do it, for better or worse, is not by understanding, I don't know, hydrodynamic turbulence, which is dear to my heart, but simply by having radars and telephones, right? So if we understand, right, if we can see if we can detect something coming and we know which way the wind is blowing, we can predict in short term with pretty good accuracy. So now the question is, with the ability to sequence, with the ability to analyze data, with the ability to communicate, what is so the population genetic equivalent of you know, the radar and the windsock? So with luck, we'll come to that. But let's start with the basics. And here my problem is, I think for some of the audience, what I'm going to say here is going to be completely trivial. But unfortunately, I need everybody to be on the same page. So I'll try to go fast. And if I'm going fast, please don't hesitate to slow me down. So obviously, evolution cannot work without heritability and without so the possibility to define again, so a heritable relation between the genotype and the phenotype. So I will very briefly talk about that. Then we'll talk about the sources of genetic variation, so mutation and sex and recombination. We already went over this in an example. And last but not least, of course, there is natural selection of the fittest. And so remarkably, so the notion of fitness is very controversial. And experts will have sort of serious discussions about it. And I'm going to be completely naive about it, and we'll stick only with sort of very intuitive definition. Okay, so we know that the genome is a long string of letters which encode amino acids, and we'll be interested in polymorphisms, which in the simplest case are just substitutions of a given letter. So if this happened to be a codon, right, so a 3 base sort of coding for amino acid, and this last G is changed to a C, actually this, this particular example doesn't change the amino acid. It's still the same serine, so we call it synonymous. But if it's the first letter that is changed, it's basically always changes amino acid that we call non-synonymous. Then there can be sort of particularly insidious mutations. In this case, if the second one is mutated to A, instead of coding for amino acid, it's going to give a signal to stop the translation. So this will basically truncate the gene, and that will most certainly have severe consequences. Synonymous thing, very naively, shouldn't have any consequence. In reality, it may have some consequences. I think Mukund already talked about sort of codon bias. Some codons are translated faster than other ones. And again, so substituting one amino acid for another may or may not be a big deal. This, of course, is not the only thing that can happen to a sequence. In the course of replication, few of letters might be dropped or repeated. So we call this deletions or insertions. So normally one would say insertions or deletions and call it indels. If such insertion or deletion involves a long stretch of DNA, the whole gene or multiple genes can be deleted or replicated. And one speaks of a copy number variation. And it actually turns out to be very important in cancer. So this copy number variation is observed in tumors. In fact, it's sort of much more widespread in tumors than even single site polymorphisms. So this was the genome. In the population, you will see these polymorphisms, variants. But even if you have a lot of them, typically there'll be a small fraction of the whole genome. Maybe a tenth of a percent. That's what it is between any pair of us in this room. And typically, you will not see a single locus, right? So a single letter in in 3 different variants. You will see only one variant. So this is kind of nice for the statistical mechanicians among us, or also computer scientists among us, because then we can actually forget this whole alphabet soup business and only refer to the presence or absence of a variant. And we have a little binary variable, and you can call it 0, 1, or plus minus 1, depending on your scientific persuasion. So back to this genotype-phenotype map. We now want to define this function on a hypercube, right? 2 to the L possible combinatoric states in the space of polymorphisms. Of course, knowing what this phenotype-genotype function is encompasses pretty much all of biology. So this is molecular genetics. And we're just going to be mathematicians about it. So it's just some function that we ultimately need to know something about, at least in the statistical sense. So, well, I just said, phenotype, what kind of phenotypes might we be interested in? Well, there are, of course, many, many attributes of an organism. And some of these attributes are pleasantly discrete. So a person may have, right, so Mendel was studying the colors of pea flowers. But we learned about this at school. We also learned at school about blue eyes, a nice discrete recessive trait. We also know that wizards and muggles are so discrete trait, although I'm not entirely sure where the experimental data comes from. So that's so Mendelian genetics that we learned about in middle school. But there are other obvious traits. For example, height. And so if we were to take a little census in this room, we have enough people that we would probably discover, I would most certainly discover, that the height distribution is more or less Gaussian. And it turns out, so this is what's called a quantitative trait, right? It is a real number as opposed to a binary bit. So height also happens to be very strongly heritable. It is strongly correlated to height of the mother and height of the father, and is basically a Gaussian centered around the mean height, well, with some small corrections. So there is a systematic differences in males and females. But in any case, it is nicely distributed around parental mean. And some 100 years ago, there was sort of a scientific war going on involving so the followers of Mendelians, who were talking about the inheritance of these discrete, quantized, binary traits, and biometricians. And biometricians were studying these quantitative traits like height or oil content of sunflower or corn or the weight of hogs and all the interesting quantitative traits that are important in agriculture and that people have been acquiring data on for 10,000 years, right? All the collective experience of animal husbandry and agriculture. And so these 2 points of view seemed very, very different. And R.A. Fisher came along and reconciled them with a very simple notion. He basically said that the difference between a quantitative trait and a discrete trait is that the quantitative trait is a linear superposition of lots of discrete traits. And each of these polymorphic loci, so from 1 to L, can be contributing positively or negatively to the height. And some of them undoubtedly make much larger contribution than the others, but still there is there are very many of them. And then basically by virtue of law of large numbers and the central limit theorem, this universal occurrence of normal distributions in these quantitative traits is explained. So far, so good. But this is actually a remarkable statement, because you look at this whole emerging body of knowledge in systems biology, and there are networks, right? We were talking about networks, and things are interacting, and in systems biology and molecular genetics, there's this great deal of complexity. Where did all complexity go? This is the simplest non-interacting parameterization of this phenotype-genotype function that one can ever conceive. And so this is fascinating, right? And there are obviously several possibilities. One is this is not the whole story, and there are additional terms here that involve pairs of loci and triplets of loci and 27 loci together, and so on. That this is just the first term, and then there are all the interactions that Fisher left for us to sort out. And yes, please. Excuse me? L, it refers to just a number of these variable loci. And we don't really know what it is. But neither did Fisher. And we still don't know. But what we know is that there are many. So I think there are actually ways, now we have a lot more data, so you can try to put a lower bound on how many loci contribute. And I think for the height, that lower bound is something like 100. Oh, H is supposed to be just the height. Are there more questions? Please. Excuse me?
You already, I guess, stressed the question of linearity. You assume you sum up, but
Yeah.
there have to be some evidence sooner or later that that's a true statement.
Well, so that's exactly what I'm talking about, right? So, right, this was Fisher's theory. It was a hypothesis, and makes predictions, and it is not a true statement in the sense that it is clearly approximate, and there are corrections, right? And now we can actually study just how good an approximation it is, and what fraction of genetic variation it captures, and how to capture the rest of it. And so the non-trivial part of the story here, of course, is we're only talking about heritable variation. There is also variation that is not heritable, right? Which is due to nutrition, which is due to all sorts of environmental effects. And the difficulty with nailing so the corrections to this simple model is that it's not too difficult to figure out how much of the variance, how much of genetic variation is captured by this additive model. But what's left is more difficult to figure out, whether it is well, what's called epistasis, interactions between different loci, or is environmental variation, or whatnot. But very interesting class of questions, and we don't know the answer. Okay, so we talked about phenotypes. Let's come back to mutation and just go over the numbers a little bit. So I gave an example of HIV. I said it was few times 10 to the minus 5. So that's on the this should be pointing in another direction. Sorry, that's on the higher. Okay, sorry about this. I was doing it this morning. Okay, that's on the upper reaches of mutation rate, which is somewhere between 10 to the minus 4 and 10 to the minus 9. So in E. coli, mutation rate is 10 to the minus 9 per base pair per generation, which if you multiply by the length of the genome, so 2 megabases in that case, works out to about 1 in 1,000 per genome per generation, right? But that number refers to E. coli wild type, whatever that might be. So wild type E. coli actually is a lab strain. So there are mutated strains of E. coli which mutate 10 or 1,000 times faster. And there is actually evidence that E. coli and other bacteria can sort of kick in into this sort of high mutation rate state under stress. The human mutation rate is 10 to the minus 8 per base pair generation, but the genome is 3 gigabases, so that works out to 30 per genome generation. Well, actually, that number includes mutations that occur in so the process of, let's see, gametogenesis, right? So there are about 30 cell cycles separating our ancestral gamete and the eggs and the sperm that we produce. So the number is more like 1 per genome generation, which if you're willing to be flexible, to work with sort of accuracy on a logarithmic scale, and if like me, you have limited memory. So one number to remember is 1 per genome, and it's actually a bit lower than that. So a tenth of a genome, I'm sorry, a tenth per genome generation is a reasonable ballpark number that will see you through most situations. And we already talked about HIV, which has a much higher mutation rate, but curiously, in terms of so the full genome, works out to be pretty comparable to what we got. Yes?
Like in E. coli, so there are some strains which are having lower mutational rate, but mutated strains are having higher.
Mm-hmm.
So is it due to only the factor of this replication, proofreading, infidelity, or there are some other factors also?
Yeah, so it is easier to increase mutation rate than to decrease it, right? Because really all you need is to turn off the proofreading. But it's an interesting question of what should mutation rate be, right? And there is this somehow notion that mutations are bad, but you can't have evolution without mutations, right? So one of the interesting fundamental questions to think about is actually trying to understand what, if you like, optimal mutation rate might there be. But of course, you have to be prepared to the fact that optimal is not necessarily terribly universal. It depends on various ecological factors, depends on population size, depends how rapidly the environment is changing. But it's generally an interesting thing to contemplate. Yes?
Related to that, Boris, is this 10 to the minus 3 figure, which I've never thought of a good reason for how things get driven low.
Yeah.
What kind of, you could say it's drift and somehow it got fixed. That it's just drift and somehow it got fixed at 10 to the minus 3 for genome. But you're only selecting on 1 out of 1,000 organisms when it's that low, right?
Yeah.
A lot of them are nuclear as well, so.
Yeah. Well, so that's an excellent question. So you have these, so if you like, parameters of evolution, right? Evolution, so mutation rate, recombination rate, and they're so tiny that the contribution to fitness in an individual, in one generation, is almost zilch, right? And what's worse is it might mostly be negative. Right? But so the question is, how do we begin to think about the evolution of these evolutionary parameters? And we clearly have to think of those things on a different time scale. They're not, we're not selecting the fittest in the next generation. So we have to ask, what is the long-term survival probability, right? And that goes back to this notion of the fitness. So the very stupid notion of fitness is, well, it's just an expected number of surviving offspring. That's a very short-term view of it. But if you ask a long-term question, right, knowing that long-term basically, only one member of any given population, I'm being sloppy here because of asexual and sexual. So let's think asexual. And in the asexual case, there'll be only one member of the population that's going to survive, if any, right? And we're going to see this on the blackboard. Right? So then the question is, what were the parameters of that individual that perhaps increased the probability of its survival? Right? And even if overall survival probability is small, we can talk about some sort of maximum likelihood. Is an individual with relatively high mutation or relatively lower mutation rate or some in-between mutation rate would be the most likely individual to survive in some varying environment ultimately, right? And I think that's the way in which we can approach these, if you like, evolution of I guess I sort of think of it as secondary traits. So these traits that do not obviously contribute to, at least do not positively contribute to fitness in the next generation. Okay. The big question is, okay, we have these mutations, but how much do they change the phenotype? And the true answer to this question is, boy, I wish I knew. And nobody really knows. And what's worse, it's not even obviously a well-posed question, because it may depend on the situation and on the bug. So, for example, for this wild type E. coli that has been living in the lab in a nice, perfect, stable environment for a very long time, well, there are probably very few mutations that it can actually do to improve its so performance, at least in the short term, in this stable environment. Right? So the probability of finding beneficial mutation is probably very, very low. But if you throw it into some unpleasant condition where it's mostly dying, now there'll be perhaps many more ways in which it can improve its sort of poor performance. So with that disclaimer, let me throw some numbers at you. So there's a...
I recently read a paper where the contradiction was kind of there was a generation of shaker flask experiment. Forget in which lab in the
Yeah.
United States. They found that even in beneficial
Yeah.
environments, there did happen
Yeah.
mutations. So that was something different there. So
I see. So this all has to be taken with a large grain of salt. But again, I like to have some crazy numbers in mind, even if they are bad numbers. You know, you can, I guess, argue whether a bad number is better than no number. I don't know. So roughly something like 10% of mutations have, you might expect would have a large effect. Let's call them lethal. Something like 10% would have a weak deleterious mutation, so will affect the rate of growth on the scale of 1%, right? That basically means it will become visible on the scale of, will become a big effect on the scale of 100 generations. And a tenth of that is would be sort of beneficial mutation rate, again, at this modest level of improvement. And you know, I make it not a small print, but a very large print, a disclaimer. Okay, so I'm not doing that well on time. I was going to tell you about so sex and recombination, but that's something that you all know about. You know, boy gamete meets girl gamete. So I'm just going to skip that. Instead, let's get actually into population genetics proper. Let's start thinking about what happens to genetic variants in a population. So let this bunch of circles, disks, this column represent a population. So there's a bunch of individuals, and for starters, let's say that they're all genetically identical. So think of this as sort of a single strain of you know, invading virus. And so perhaps this is happening in the presence of a drug, so they're not doing terribly well, none of them. But then one of them acquired a beneficial mutation, and it started growing on average more rapidly than its brethren, and eventually it took over the population. Of course, even beneficial mutations are not guaranteed to succeed, because just by chance they can become extinct. So what we want to understand is actually the probability for a new variant, whether beneficial or effectively neutral, right, not having any fitness advantage or disadvantage, or deleterious, for that matter. We want to know what's the probability to become extinct, and the opposite, what's the probability to take over the population. So in the jargon of population genetics, so this process of one genotype. There is an escape mutation there. This was supposed to be green. What happened? I don't know. So, okay, this is the new mutation coming in. But before it came in, the green genotype has reached fixation. And what we'll want is to actually describe quantitatively these probabilities. And we're going to do this on the blackboard, but maybe I'll just advertise the results. So first, one wants to consider the stochastic fate of even neutral mutation. And that was worked out by Kimura in the 50s. And the logic was basically the following. The thought was that, okay, beneficial mutations, right, grow in number, grow in frequency, and fixate. So if that happens rapidly enough, okay, if they come into population not too often and fixate rapidly, you're fairly unlikely to catch one in the act. Okay? On the other hand, if mutation is strongly deleterious, it's going to lose out in the race, and again, you're not going to find it in population. So then this notion of neutral theory was that all of the variation that you see in the population is essentially neutral. And those are the alleles that are eventually on the way out of the population. They're on the way towards extinction, except for a few that fixate. And one can actually calculate this. And we're going to do exactly that. One will find that, okay, if a new allele comes in at time t0, then t generations later, its extinction probability is approaching 1, right, as 1 over the number of generations. So one can actually calculate more and calculate the probability of finding exactly n, let's say in this case, exactly 3 alleles in the whole population. And that is given by a simple little formula. This probability is exponential in the number of copies. Right, has this 1 over time, just like the Boltzmann factor. So this is basically saying that so the characteristic number of copies that you may find in the population, provided that the allele is not extinct, increases linearly with time. It also means that in this neutral case, there is a finite probability for the number of copies to become equal to the population size, which is to say the allele will fixate. So if this little n is to become this large capital N, the population size, right? Large N over T will only become probable here if T itself is of order of the size of the population. So one can actually demonstrate this explicitly. So the time to fixation is scales linearly with the population size. And, but of course, in order to fixate, the allele has to survive for that long. And this little formula tells us that survival probability, right, is 1 minus the extinction probability. So 1 over time. Time is n. So fixation probability is 1 over n. So these are sort of Kimura's results. Now with for the rate of adaptation. And let's first start with a situation where mutation rate is low and the population is not very large. And so if we're interested in adaptation, we're particularly looking for beneficial mutations. And those we said are less frequent than deleterious mutations. So deleterious mutations are going to come in and will be selected against. But this beneficial mutation will appear every so often. And if it does not become extinct right away, it will sweep through the whole population, will fixate. And then the population will wait for another beneficial mutation come in. And if it does, it will incorporate it in the same manner. And you can ask, how does the, what is the rate of increase in the fitness? And it's going to be proportional to the rate with which mutations are coming into the population, which is mutation rate times population size. And each event like this is going to bring in a factor of S. That's, sorry, I should have defined it on the slide. So S here stands for this additive contribution to the fitness. It's positive. And another factor of S is the survival probability for that beneficial mutation, right? We just talked about even beneficial mutations when they're present in only a very small number of individuals are susceptible to being becoming extinct by chance. Okay. So far, so good. So next, let's think about what happens when, in a situation where the mutation rate is higher or the population is larger. So now, we basically turned up the faucet of mutation rate, and these beneficial mutations are coming in more rapidly. And now, you expect them to occur in different individuals simultaneously. So some will arise before the previous ones fixate in the population. But now in the sexual population, only one of these guys can take over. So the rest are going to be lost. Yeah, yeah, effectively, yes. Well, so the relevant thing is not quite 1, but some logarithm of n. But it's smaller than something, so sufficiently small. And when it gets to be, right, because it, you know, the relevant time is so how long it takes to fixate. And that's logarithmic in population size. But, you know, log n can get too large. Well, and that's sort of the whole point, because it turns out that because most of the mutations are lost in this case, the rate of evolution is no longer linear in mutation influx, but instead depends only logarithmically on n. And this is actually a remarkable bit of sociology or whatever, if not science, history of science. These pictures I pulled out from a rather old textbook. So this was, on some level, realized a long time ago. But so the quantitative understanding of what this behavior is actually remarkably recent. And we are going to derive this result tomorrow. Well, so, right, obviously, right, for, you know, provided that n mu is sufficiently large, so obviously the logarithm is much smaller than what this number would have been otherwise. So wouldn't it be nice to be able to regain some of those lost beneficial mutations and accelerate evolution? And that's, again, according to the old textbook, is the benefit of sex. So the idea here is that in the presence of recombination, there is a possibility that beneficial mutations that arise on, you know, different individuals will be brought together onto one gamete, and will both have a chance to contribute to fitness together, right? And now in this happy family, there is no mutations are lost, no beneficial mutations are lost. All of them are incorporated into the future of the population. And this conventional wisdom would then basically say that with the benefit of sex, one recovers this linear dependence of the rate of adaptation on the influx of mutations. So obviously, this makes a difference if, okay, I'm sorry. So who cares about you know, n's or log n's? But it actually makes a difference if you're trying to think about the rate of acquisition of drug resistance in HIV, right? So the question there is, okay, there are drugs, and these drugs can suppress the viral load. Cannot completely eliminate the virus, but they can suppress it. Now the question is, how far should it be suppressed, right? What is the gain per factor of 2 in sort of residual viral population size? And since in the case of HIV, recombination is part of the evolutionary game, this question is relevant. So as I was saying before, with Kimura's theory, the standing paradigm in population genetics for a long time has been this kind of a picture, that most of the standing genetic variation that one observes in the population doesn't matter, that it is neutral. And the things that matter occur seldom enough that we only need to worry about them one thing at a time. And we've understood, or for that matter, the founding fathers have understood what's happening in this situation very well, cold. But then it turns out that where it comes to the situation of non-neutral genetic diversity, we still don't understand very much. Or maybe I should say that maybe we're beginning to understand a fair bit. We're beginning to make progress, but this progress is sort of fairly recent, let's say last 15 years and maybe less. And that's what we want to understand. Okay, so you may say, well, he's interested in this interacting system because he's a statistical physicist, and it's an interesting thing to study. But it is completely irrelevant to, in nature. And what we're learning now is that the sort of situation corresponding to large population, rapid mutation rate, standing genetic diversity is more of a rule than an exception. And the beauty is that now one can do experiments and certainly determine this from sequencing, but in the case of this experiment done at Princeton not so long ago, I guess it was done before that, but it took a few years to get published. One can actually engineer yeast in such a way that certain mutations become literally visible, as in fluorescent. And so, of course, it's just one particular class of mutations that become visible. But it's a sufficiently large class that actually turns out that pretty much any population over experimentally accessible number of generations is going to generate such mutants. And then one can basically experimentally obtain the kind of diagrams that I was showing you as cartoons drawn by hand. So here a mutant emerges and fixates with the population. Here a mutant comes in and disappears, basically being displaced by some other mutant that has outcompeted it, and so on and so forth. And as I say, there are even better ways of tracking the situation by tagging yeast genetically and then sequencing. And then one can actually see the expansion of certain clones at the expense of the others, and one can document this clonal competition going on in the population. So what's going on is there's genetic diversity, and this diversity is not neutral. These guys are battling it out with each other. So that's basically the agenda. We'd like to obtain this general description of natural selection in highly diverse populations. And if you like, if anything, use that description to replace the neutral theory as the null model. So right now, if you don't know anything about interpreting genetic data obtained in the population, you will assume that it is neutral. And I just argued that that probably is not a good assumption. Well, not probably. Certainly is not a good assumption. So the question is there something that we can replace it by? So short of knowing the effect of each and every possible substitution, which we never will have, right? With all the molecular genetics effort, we'll never know everything about that gigantic 2 to the L hypercube. So, but perhaps even in the absence of this complete molecular genetic knowledge, and in the context of for these evolutionary dynamics questions, one can obtain this sort of statistical description in terms of, as I was saying, the frequency of beneficial mutations, the rate of evolution, and so things that make sense, even if you don't know each and every mutation effect by name. And that is what we mean by statistical genetics. Now, just like statistical mechanics. That was the my little introduction, which I thought to go through in half of this time. Okay. I did a lot of talking, and I'm getting a little tired of talking. But I'd really like to calculate something. Okay. Yeah. Yes. So, okay, it would take me a little too long to actually explain what. Oh, so yeah. So this is just a cartoon. So you're probably thinking that this doesn't look like that. It doesn't. This was a cartoon, and this is sort of many examples of this sort of behavior, where some fraction of the population, in this case from the very beginning of the trace, was fluorescent, right? And this is done literally by flow cytometry. Right, you run the cells and look what fraction of them are yellow, right? And you do this many times, right, as a function as the time progresses. And so in this case, so for the longest time there weren't any yellow cells, and then they appeared, and shortly thereafter took over the population. And this is actually quantitative information. So from fitting exponentials to the slope, you can estimate the fitness advantage of that particular mutation. And you actually right? So you collect enough of this data, and you can start sort of reconstructing the probability distribution of mutation with this or that fitness advantage. So that's a very interesting experiment. But then actually more often than not, one doesn't see the simple picture. Instead, right, this mutation comes in, and then the yellow fraction of the population poops out. Right? Did I answer the question? Okay.
So the problem when I look
Yep.
at the data, Boris, is that some of those columns look the same, yet there's different things attributed to them. The D column doesn't look like clonal interference to me at all, except for the third panel down. I would have thought that, oh wait, there's different mutations that appear there too.
Yeah. So you're absolutely right. So, and how about this, right?
Like what I call
Frequency dependent.
No, I
Yeah.
see that as clonal interference, except it's not. They're both either selectively neutral or their W values exactly the same.
Well, one possibility is that this is some frequency-dependent mutation, and it has came through to reached whatever 30% of the population and started interfering with itself for some biochemical reason. And well, anyway, it's a zoo.
Could I ask another kind of
Please.
question? So, in an attempt to come up with a kind of a stat-bank way of reformulating these things, which would be extremely interesting at least, I've always thought that somehow you have to put in things like fluctuations in the environment. Like you actually have to have explicitly some function that says that there's a periodicity, or it's so noisy there's no, that kind of thing. But do people do much of that? I mean,
Well, people are beginning to do that. That is, so excellent question, right? And okay, you know what? I'm going to do the following. I'm not going to calculate anything today. I'm going to calculate tomorrow. And
Time, though, if you want to take a break now and start again at 4, you're choice, though.
okay, but I think everybody's tired. I'm tired. Everybody's tired. I can do it after a break.
Half an hour break. So, because we had this tutorial slot, so in the interest of continuity, we could, if you think by not doing this, you won't get to something later.
Listen, it's all flexible. It's all flexible. I think. But sorry, I interrupted. So a good question and interrupted the bad answer. So let me.
do you see a way to make progress?
Absolutely. And in fact, so I was optimistically saying that we collectively made some headway in the field, and that's actually where many of us are headed, right? But so at the same time, right? There's time and there's space, right? Another very important thing is, better than me, is that selection pressure is not necessarily the same everywhere in space. So right now we're talking about this single chunk of the population subject to the same selective pressure. But instead it could be, and it's not could be, it must be, it is, many different colonies, many different islands that are subject to a little more antibiotic or a little less antibiotic. And then the probability for some mutation to establish itself in the presence of less antibiotic is higher than in the presence of a lot of antibiotic, right? And then that mutation can, by wind or birds or whatever, spread to another little patch. And this, so spatiotemporal, right? So I was talking about this fitness landscape being a big hypercube. So now take that hypercube and make it time dependent and space dependent. So that's basically the, if you like, ecological landscape that evolution unfolds in, right? And the question is, how do we manage to make even statistical statements about it? And so the notion of fitness gives out, right? So that's sort of why we can argue about the fitness of a given genotype. It depends on the environment. It depends on how well adapted it is. I'm sorry. The effect of a given mutation, not a given genotype, the effect of a given mutation depends on the environment that this genotype is subject to. But it still makes sense to talk about long-term survival. So talking about the extinction probability, these notions that we touched upon here, are very powerful. So that is really the, I'd say, correct way of thinking about fitness. It's not what, so the fitness today, it's the survival in the long term that matters, right? And then we can at least start thinking about, so calculating the survival probability in a time-dependent environment. If you can sort of sensibly parameterize this time dependence, saying that, well, it changes on a certain time scale and it doesn't change by a lot, so you will then say that the fitness itself is diffusing in some crazy space, and you can start parameterizing it, right? Well, all of that is work in progress. What questions? So something is very wrong here. I think they're tired.
So we start again tomorrow with
Okay, I think, yeah, I think we'll do that.